Glucose enters the cell and starts being broken down in a process called glycolysis, which consumes two ATP molecules. This creates two pyruvates, two water molecules, two NADHs, two hydrogen ions, and four ATP. To make ATP, adenosine diphosphate has to bond with a phosphate group in a process called phosphorylation. More specifically, it's substrate level phosphorylation because enzymes do most of the bonding here. To make NADH, NAD plus gains two electrons plus a hydrogen ion. Entering a mitochondrion, pyruvate is robbed of electrons to make NADH, stripped of one carbon dioxide molecule, and bonds with coenzyme A to make acetyl-CoA. Acetyl-CoA goes on to power the Krebs cycle, which takes two acetyl-CoA molecules and spins around twice to break glucose into two carbon dioxides, six NADHs, two FADH2s, and two ATPs from substrate-level phosphorylation. Most of the energy produced so far is stored in the electron carriers and go towards the final step called oxidative phosphorylation. Traveling to the inner membrane of the mitochondria, the electron carriers offload their cargo into proteins that use portions of the energy to pump protons into the intermembrane space, the area between the outer mitochondrial membrane and the inner mitochondrial membrane, to make a proton gradient. This electron transport chain is only one of thousands on the surface of the inner mitochondrial membrane, which is folded up in structure called cristae, and at the end of it the electrons bond with two hydrogen ions and oxygen to make water, opening up the chain for more electrons. The pumped protons proceed to pass through a protein called ATP synthase, causing ADP to be phosphorylated into ATP much more efficiently in a process called chemiosmosis. Of the roughly 34 molecules of ATP made in cellular respiration, oxidative phosphorylation accounts for about 30 of them. Fermentation is what the cell does when oxygen is not available. It all has to take place in the cytoplasm and is an extension of glycolysis. Alcohol fermentation makes ATP through substrate level phosphorylation of ADP and keeps itself going by oxidizing NADH so that it can recycle NAD plus molecules as byproducts or CO2 and ethyl alcohol. Lactic acid fermentation also makes ATP through substrate level phosphorylation of ADP and keeps itself going by reducing pyruvates to turn NADH into NAD+. Its main byproduct is lactic acid.